My name is Alexander Schmitz. I'm a PhD student in the University of Frankfurt here, and I'm a bioinformatician since I started in 2006 with studying bioinformatics. And what I try to do with Mathematica is in principle, I try to help all the biologists which are in our group with visualizing and analyzing their data. And what I apply Mathematica currently to is to study lateral root development in plants, and we tried that with cell nuclei tracking, where we have developed programs with Mathematica, and then we try to analyze that data. So what I would like to show you in the next couple of minutes is how we use Mathematica in our group to extract and analyze important biological information from 3D image data. And the main topic I'd like to focus on is to do cell tracking in the development of lateral roots in a plant called Arabidopsis thaliana. So I work in a group which is called Physical Biology Group, and our group mainly consists of a quite diverse set of people. So we are biologists, chemists, physicists, we also have mathematicians, and also me as a bioinformatician. And we try in our group to combine several aspects of cell and developmental biology together with 3D time-lapse microscopy. So if you have heard the talk by Ernst Stelzer, you might know a little bit about that now. We also do data processing in our group and mathematical modeling. And in general, if you are in the life sciences, you have to go through a certain series of steps. So you, in principle, start off with some scientific questions. So maybe you want to understand a certain biological process. So in principle, as I said, we have a certain biological question we want to address. Therefore, we in our group develop the methods, do the experiments, acquire some image data with the microscopes. Then in our group process the data, analyze it, and derive some kind of a model which then can be used to explain how this biological process how we can understand this biological process. And as a biologist, as a bioinformatician, I'm mostly involved in these steps. So I'm mainly involved in data processing, so image processing mainly, and data analysis. And many of us in our group apply Mathematica on a quite regular basis. So for me, I apply it on a daily basis. And therefore, we also provide introductory and advanced Mathematica courses. So with that said, I come back to the topic. So we in our group try to investigate how plant roots really branch. So how are lateral roots formed, and how does this process really work? And for that, we set up plants in a microscope under very close to natural conditions, and record the whole process as a time series. So you can think of this whole process, if you're not familiar with microscopy, that doesn't matter, but you have somewhere your plant in a microscope which sits in front of an objective lens, and then this whole process is recorded via fluorescence microscopy. But what you get out is a recording like this here on the right-hand side, where you have the main root, so that's the main plant root, and here's a lateral root forming at the side of this plant. So as you can see, this takes quite a while, so this recording is over several days, and you also see that certain biological structures are visible. So for example, you can see in this recording here these red bouncing fancy balls. Those are cell nuclei, if you're familiar with that. And in order to understand now how this process really works, how this formation takes place, we in our group tried to track these cell nuclei and investigate what is the contribution of single cells to this whole process. And therefore, we developed Mathematica programs. So I would like to show you now the main program we use for the tracking. And first of all, I have to import a package where I have the main function. So if I open that, It's a little bit small now. Okay. That's the main notebook. So I have created a notebook now. And in principle, first of all, you see here on the top left corner, there are some action menus which can be used to import and export data and apply some options. And let's first of all import a time series of such a recording. So I go to Open Time Series and go to a folder where my data is located. And what the program now did is it imported the first stack of the recording. So if I go now with the controls here through this plane slider, we're basically moving plane by plane through the 3D volume. And there again you see the cell nuclei. So here are the structures which evolve to later on form a lateral root. And we also can go through time. So this will load the next time step of the recording, which in principle is again a 3D volume. So what we can do now here in the program is to track individual events or individual cells. And what I also use quite heavily in this program is this magnification here, for example, which basically dynamically shows you a magnification of the position in the current image. So if I apply a little bit more magnification and move a little bit deeper into the stack, there you see on the left-hand side in the magnification that here is a cell which is outlined with this green line here, and in the center there's a red nucleus. So this is the object we are interested in. And if we go through time now, you can see that in the next time point, this red object seems to split up into two new objects. So in biology, this is called a cell division. So basically what happens is that this cell now splits up into two new cells. And we want to track all of these events. So what we can do now in the program is we click on the position of the cell, and this will create via event handler. It will store the information from the mouse position and all the stuff. And what the program then stores is all the information like the object ID, which is generated automatically. We have the positional information like X, Y, and Z, and we have the time point, and another list, which is the precursor list, where it now stores the ancestry of a certain object. So if this object now derived from another object, then this list will contain a value. So let's track now the second event. So we have this cell division. Let's go back and we can select now this object and say start a new track from my selection. And if we go now one time step further, we can now highlight this object, and there you see it, besides the positional information, it also stores that this object derived from the previous object 1. So by this we have not only marked the position of the cell nuclei, but we also have some kind of lineage information which shows us which cells derive from which objects. And we basically did this tracking with several recordings of lateral root formation. And what we can do then, if we are finished with all the tracking, we can go to Save Objects, to Excel file, and this will now export Microsoft Excel file where all the object information is located in. So what this looks like, if I go back to the presentation, I can show you in a second, is if I import now this Excel sheet, then you can see that in principle there only the information of all the objects is stored. So again we have the object ID, several information about the position, and we also have this precursor list. And this precursor list, which shows the lineage information, can also be a little bit more complicated, as you can see here. So this list has now 2 entries, which just means that object number 340 derived from 248 and is derived from 219, which is, in this case, an initial cell. So what we can do now with this information is, first of all, play a little bit around and extract certain features. But what I want to show you now is a more specialized way of importing the data. So I have here a function which basically converts all the stuff which is in the Excel sheet to a more rule-based representation. So if you have seen the talk by Tom Wickham-Jones in this morning, he talked about this association. in principle, I'm doing the same thing with rules. So if I import now the data and display the first object, then you see that objects are now represented as a set of rules where we can directly access, for example, a certain element. If we want to know, for example, what is the precursor information of that object list at position 250, for example, precursors, then we directly get all the precursor lists. So we don't need to know about where this information is stored, so I don't need to know the position or anything else. In principle, I just use a rule replacement to extract the data. So with that, remember that we also have the ancestry or the lineage information stored in this list. We can now visualize and analyze the whole lineage information of this. And what I use here is I basically use graph, so the built-in functions of graph to do that. Sorry. It doesn't show. And what I first of all do is I try to sort all the objects in a way such that they all belong to the same lineage. So each object that belongs to the same lineage will be in one list. And if we then try to convert this into a more obvious representation, we can use graph to create such a tree-like structure where we have all this lineage information in one object representation. So as you can see here, still the object information is there. I just put it into the vertices, and the edges now hold the information about the time domain. So for example, we have here this object number 4, divided after 4 time points, split it up into object 19 and 20. So basically all the information is now in this representation here, and we can start visualizing certain aspects of that. And what I do is I basically use the function setProperty and property value again to extract information. So if we want to know a certain feature of a vertex in this tree, we can use property value to extract the information again. What I would like to show you in the end is now a visualization of the information we extracted from this recording in just one simple manipulate. So what I have here is I have done the tracking of nuclei positions in several recordings, and I create a manipulate now where the object locations are displayed as colored spheres. So each colored sphere here is basically the position of one nucleus, and the colors represent different information, but still we have all the information about the object there. So this is now displayed via a tooltip, and now what we can do is we can start colorizing the objects according to different features. What I have done here is I have 3 data sets which I can select now here. I have a time domain slider, so in principle go through the time domain, and I can now visualize certain features. So if I look now at the feature lineage, we can see how this structure evolves over time. So this is a nice way to directly access different parts of all this huge information. And we can also restrict the visualization to certain areas, for example. So with that, I would like to summarize this part. So I've shown you a way of applying Mathematica in studying root branching in plants. I showed you how we use Mathematica to create simple user interfaces to do, in this case, 3D cell tracking. I've shown you a way how you can store data, so store it in Excel sheets and import it again in a rule fashion, and then visualize the data, for example, as a graph structure. And I showed you how you can use manipulate in order to visualize and analyze multidimensional data sets. So with that, I would like to thank my group, especially Ernst Stelzer, who's supervising me, and also especially Daniel von Wangenheim, who's the main biologist in our group, who's doing all these experiments and all the recordings, and especially also my dear colleagues, especially Sabine and Bina. And in the end, I would also like to thank you for your attention, and I'm happy to answer your questions.